Thank you, Pete, for the very nice introduction. I've been in pathology, practicing pathology and laboratory medicine for longer than I'd care to admit, for many, many years, and have seen a lot of changes in that time. But just to give you an idea of how long ago I started out, when I started in lab medicine, there was no such thing as flow cytometry. There was no such thing as immunohistochemistry. There was no such thing as a handheld device that could measure electrolytes and so forth. We did have running water, though. So I've seen a lot of changes over that time, obviously. Technological, reimbursement, changes in regulation. But I must admit, the current environment is unique to me, in my experience, with the number of changes happening and the impact that those changes may have on the practice of pathology and laboratory medicine. And I've just listed some of them here, which I'm sure you're familiar with. And but I elected to concentrate on one area for this talk, and that is in the area of regulation, and specifically the regulation of laboratory-developed tests. Now, that may not sound like a very big deal, but as a matter of fact, it's something of great concern to the CAP. The CAP is very active now, trying to find ways to better support pathologists, pathology, and laboratory medicine in this ever-changing environment. And we may be over-regulated in America, but there's no question that laboratory-developed tests are going to be subject to more regulations in the future, and such that CAP has designated this issue as a, quote, top quadrant risk. What that means is that we at CAP have a short list, a grid of those factors which we consider to be the highest risk to the practice of pathology and lab medicine in the next few years. There's only 9 of them, actually, and one of them is related to laboratory-developed tests. And to paraphrase what it says in the grid, laboratory-developed tests, as the result of new regulations, pathologists and laboratories could experience increased costs and time frames for development of new tests, thereby stifling innovation and limiting our ability to develop a test within the laboratory. And I think you'll see during the course of this presentation that that's not an overstatement. Now, the first thing we actually need to do is talk about what is a definition of a laboratory-developed test. It would seem obvious what that means, but there actually is a specific definition that the regulators and accreditors look at, and it's up on the screen now. And that is, a test is obviously developed by a clinical laboratory, but it is performed only in that laboratory. In other words, if the test is given to another laboratory to perform, then it's no longer an LDT under this definition. Also, by definition, the test is not FDA approved or cleared, and there's a grandfathering date here also, and I can, if you're interested, I can tell you where that date came from. That date is a CAP date, by the way. I don't know if the regulators are going to accept that. Now, we're going to be talking a bit about the FDA today, which is kind of it's a new area for a lot of us. Except for blood bank, we don't really think of FDA being involved in laboratory testing per se. But at this point, I just want to mention that the difference between an FDA cleared and an FDA approved test. An FDA cleared test is a test that a manufacturer puts out on the market, but there is already a test out there which is similar. So the manufacturer just has to show that the new test has similar performance parameters to the old test. So that's cleared. An FDA approved test, on the other hand, is one which has no predicate device. That's FDA speak for previous test. Has no predicate device, and so the manufacturer has to establish all the specifications for that test independently, and that's a more onerous regulatory process. So when I'm going to use FDA cleared, FDA approved sort of synonymously, but they're two slightly different things. So the way this talk is organized, first I'd like to review what the current regulatory and CAP accreditation requirements are for lab-developed tests. Then we're going to talk about the rationale, the kind of thinking why the FDA wants to get involved with a direct regulation of tests that we develop in our own laboratories. And I want to illustrate this with some recent FDA approaches where it's sort of the FDA's communications are kind of nibbling around the edge of the universe of laboratory developed tests. It kind of gives us an idea for what FDA is thinking. And then finally, CAP proposal for a joint CAP-FDA program that would hopefully not put an onerous burden on laboratories in the development of tests and yet would also satisfy the FDA in terms of regulation. So what are the current requirements? And really what we're talking about in terms of the requirements are the requirements to validate the test. Validate a test such that it's acceptable to use that test for patient testing. Currently, as you know, laboratory tests are regulated through CMS, the Centers for Medicare and Medicaid Services, through CLIA, the Clinical Laboratory Improvement Amendments of 1988. The FDA is not involved in that except in a few very peripheral ways, which I will mention. And accreditation, of course, I'll talk about the CAP accreditation program. There are others out there, obviously, but I'll concentrate on CAP. Now, where do laboratory developed tests fit in the CLIA test classification scheme? As you probably know, there are two broad classifications of testing under CLIA. The first is wave tests, which are tests which were considered, at least the original idea was that they were so simple that they would not have to be subject to the detailed CLIA regulations. So that's the first category. The second category are the non-wave tests, which is everything else, which all the other tests which are subject to the detailed CLIA regulations. And the non-wave tests are further subdivided into tests of moderate and high complexity. And the difference in CLIA between moderate and high complexity is mainly in the personnel requirements to perform the test, to direct a laboratory that does the testing. In CAP, there is very little difference, okay, between moderate and high complexity testing. Now, by definition, essentially all laboratory developed tests are high complexity because any test that's not almost any test that's not FDA cleared or approved is by definition a high complexity test. So that's where LDTs fall into this classification scheme. Now there are three elements we can look at in terms of the current regulatory climate in the regulation of laboratory developed tests: analytic validity, clinical validity, and clinical utility. In this context, analytic validity means the test's ability to accurately and reliably measure the analyte of interest in the patient population of interest. And where the analytic validation requirements are in CLIA, they mainly reside in this section, which you see listed up here on the board. And what CLIA delineates are 6 performance specifications that laboratories must address in validating a test. And those are analytic accuracy and precision, reportable range, reference range, interferences, and lower limit of detection. But CLIA requires a different level of validation depending on whether the test is FDA cleared or approved or not. For a test that's FDA cleared or approved, all the laboratory has to do is verify the manufacturer data on accuracy, precision, reportable range, and reference range. But for laboratory developed tests, the laboratory has to independently establish the data for all of those elements. Now, CAP is very similar to CLIA, so the differences are minimalistic. So I won't go into that in detail. One thing I will mention is that the CAP analytic validation requirements in the past were in the laboratory general checklist. They've been moved to a new checklist. We have to keep everybody guessing, of course, so we keep moving things around. They're moved to a new checklist called the common checklist, which is so named because it contains checklist items which are common to every lab discipline. So they've kind of been consolidated into that checklist, actually thereby reducing the total number of checklist requirements. So that's where analytic validation will be in the future. And one point, we get asked about this, so I want to make one point that is mentioned in the checklist, which is that obviously not all tests are subject to all of the method performance specifications. Clearly a qualitative test, you're not going to have the same issues with the reportable range and precision that you would with a quantitative test. Now, one point is that neither CLIA nor CAP have any specificity in terms of requiring a minimum number of samples for establishing, for instance, analytic accuracy or any of the other specifications, or the minimal number of days over which a study has to be performed, or the acceptability limits for your accuracy or precision. We talked about that at CAP a number of years ago, but decided that there was just so much variation in different tests and different patient populations. Is a test being used for diagnosis, monitoring? Is it being used for screening? That you really can't come up with a generic set of numbers for that. So that's never been done. There is one exception, however, in CAP, as you probably know, and that is in breast carcinoma, for HER2, estrogen, and progesterone receptor testing. For those tests, there are some minimal numbers for the performance specifications. And I have a feeling, I don't know this, but I think that in the future, probably to address some of the concerns by FDA and others about laboratory-developed tests, especially molecular genetic tests, that we may need to put in some specific numbers for validation of that testing also. But that's just my feeling at this point. Nothing has happened with that. There are, of course, other elements of analytic validation, which I've listed here, which are scattered throughout the CAP checklist and in CLIA, and those are fairly straightforward. Now, clinical validity, the second element, in this context is the ability of a test to predict the presence of a disease or risk of a disease using measures such as sensitivity, diagnostic specificity, predictive value of a positive or negative result, and so forth. And the first question is, what are the CLIA requirements for clinical validation? The answer is there are none. CLIA says nothing about the clinical validation of a test. CAP does have some clinical validation requirements, but they are confined to the molecular pathology checklist, so they only apply to molecular pathology. And the reason for that I'll get into in a minute. But the fundamental requirement is this checklist item, which you see here. And the note to this checklist item makes the point that ideally the laboratory would do its own in-house studies on clinical validity, but recognizing that this may not always be possible or practical, and for a very rare condition, there may not be an adequate number of samples, so the laboratory can rely on literature for that data. And conversely, for a very common disorder in which the clinical validity is well established, the laboratory can rely on the literature. There are a few other clinical validation requirements in the CAP checklist. This one requires that the laboratory summarize, for laboratory-developed tests, the laboratory summarize in its report the analytic and clinical performance characteristics of the test. Then there are two other items which were added in 2011, which is the latest edition of the checklist. The first is that all clinical claims made by a laboratory about a laboratory-developed test are validated. Now, this would at first blush seem to be redundant, but it's actually aimed at a specific situation, which is that commercial laboratories that have commercial organizations, companies that have a single laboratory in which they have a laboratory-developed test, often molecular genetics test, which is offered perhaps to the public, to physicians throughout the country, may make claims about the diagnostic power of that test, the specificity of that test, the predictive value of that test on their website, in product literature they send out, on their report. And the important point is those claims need to be substantiated by data, because in some cases that has not been true. So this requirement is that a laboratory doesn't have to make a claim, but if it makes a claim which is available to outsiders, then that claim has to be supported either in the literature or through studies done within the laboratory. And the final requirement in CAP, which was added last year, is simply that the laboratory have a list of its laboratory-developed tests which were put online in the last 2 years. The reason for that is those are the tests to which the inspector's attention will be directed most intensively, looking at the validation data, the most recently added tests. Now, why did CAP restrict clinical validity requirements to the molecular pathology checklist? Well, the first reason is that most of the issues that the public and the regulators see with laboratory-developed tests involve molecular genetic testing. But more importantly, had we applied the clinical validity requirements to the entire laboratory, we would kind of inadvertently entrap many traditional laboratory-developed tests, which have been around for years and for which clinical validation is irrelevant or would be onerous or is simply a waste of time. Things like markers used in flow cytometry, reagents used in coagulation. The conventional pap smear is not FDA approved. That's a lab-developed test. We're not going to make the lab do clinical validity studies for the pap smear. Conventional microbiology cultures are basically our lab-developed tests. Immunohistochemistry antibodies. So we didn't want to get these tests involved in that. So for that reason, the clinical validity requirements remain just in the molecular pathology checklist. Now, this is going to come back as an issue in terms of the future possible regulation of lab-developed tests, as we'll see in a few minutes. And the last element of validation, which I'll just mention briefly, is clinical utility. The net balance of risks and benefits associated with using a test in a specific clinical setting. That basically is so fraught with controversy and subjectivity and complexity that it has not been brought into the picture at this point. So that's CAP has no requirements related to clinical utility, and I don't see the regulators actually putting in any requirements in the near future. In the long term, that's another matter. So that's the current situation, the regulation and accreditation of laboratory-developed tests. Well, in the middle of the year 2010, FDA convened a conference of many stakeholders in which FDA announced, actually they'd made the announcement somewhat earlier, that it was going to directly regulate laboratory-developed tests. This may come as a surprise to many of us because we think of the FDA regulating, except for the blood bank, we don't really think of the FDA directly regulating a laboratory. But FDA has asserted that laboratory tests are actually medical devices, which is FDA speak. And I'm learning a whole new language here, by the way. FDA speak, as any of you know who may have been involved in it, is different from CLIA speak or any other laboratory language. Medical devices actually include laboratory tests. So if you look at the medical device amendments to the original Food, Drug, and Cosmetic Act, indeed it does state, if you see here, a medical device includes an in vitro reagent or other similar or related article intended for use in the diagnosis of disease or in the cure, mitigation, treatment, or prevention of disease in man. Pretty all-inclusive. Now, in the past, FDA has exercised what it calls enforcement discretion, which means FDA can choose not to enforce certain aspects of the regulations. And this does not in any way mean that FDA says we don't have the right to do this, to regulate this area. It simply means that FDA has chosen not to enforce the regulation because it considers that the risk to patients is not high enough for it to expend resources doing so. So what FDA has decided to do basically is end enforcement discretion for laboratory-developed tests. And FDA's rationale, and in some ways it makes some sense. There's been a tremendous change in the nature of laboratory-developed testing in the last decade or so. It used to be that laboratory-developed tests were confined to those tests that were developed in academic centers or in hospitals or in pathology labs and just serviced the local patient population. But now, many companies, many driven by venture capital, are choosing the laboratory-developed test route as the way to market a test. Rather than make a test kit and sell it to laboratories, they establish a laboratory, do the test in their own lab, and sell that test to clients, to patients, to physicians throughout the country. And the laboratory, these tests are much more complex than laboratory-developed tests were in the old days, are being aggressively marketed, and FDA states that the public needs assurances that these diagnostic tests are sound and reliable. Now, this last bullet raises an important point, which you'll see over and over, which FDA, and this is my own perception, FDA seems to not recognize the existence of CLIA or CAP accreditation or any other accreditation. Like these tests are being done in a vacuum with no regulation at all, no accreditation. You see this repeatedly, this kind of comment from FDA. Kind of an interesting thing, but that's where they're coming from on this. They really don't view our current processes as being particularly effective. And they may need to be improved, that's true, but the FDA doesn't think they're particularly effective. So what FDA is saying is that there is an uneven playing field for IVD manufacturers, which is in vitro devices, in other words, makers of tests, because those who choose the route through laboratory developed testing don't have to jump through the hoops of getting FDA clearance or approval. And FDA also pointed out that once the test is out in the field, there are no post-marketing recall requirements. So once the test is out there, it's sort of free-for-all. That's actually not quite true. The CAP checklists do contain a checklist item about reporting test problems to FDA, which FDA asked us to put in there, and we did a number of years ago, but they don't seem to recognize it. Now, needless to say, manufacturers have put a lot of pressure on the FDA in this direction. Genentech, for example, sent FDA a 30-page letter about 3 or 4 years ago talking about some of the points I just mentioned. And this is an abstract from that letter where they're pointing out that they want a level playing field. Now, it's interesting that, of course, the laboratory industry has felt the opposite, but there actually is a justification, a legal argument for the FDA not having jurisdiction over laboratory developed tests. And it is that it's kind of convoluted, but as my lawyer explained to me, the FDA's authority rests on interstate commerce, that all these tests that it regulates are involved with interstate commerce. If you do a test, develop a test in one laboratory and just serve the patients in your immediate area, you are not involved in interstate commerce, so therefore FDA should not have jurisdiction over that test. Now, FDA has come back and said, well, the components of those laboratory developed tests probably did move through interstate commerce, so we do have jurisdiction. The counter-argument to that is that the test is a separate entity from, these lawyers amaze me. I don't know how they do this every day. The test is a separate component, separate from its components, and so that it is not subject to FDA jurisdiction. Well, this is an argument, but I can tell you that no laboratory, no organization, certainly not the CAP, has chosen to challenge FDA in court over this. The outcome of such a challenge would be highly uncertain, and it would probably ruin your relationship with FDA were you to pursue it. So nobody's interested in doing that. So the bottom line is, I think, with the exception of a couple of bumps that I'll talk about later, I think FDA has clear sailing here toward regulating laboratory developed tests, and it's going to happen. So what I want to do now is move and talk about, very briefly, about 4 guidance documents that the FDA has put out, because these kind of show what FDA's thinking is in terms of how it's going to approach laboratories with involving laboratory developed tests. Now, these guidance documents, each one has this paragraph, which you see in the bottom of the slide here, which is that these documents do not establish legally enforceable responsibilities. They describe the agency's current thinking on a topic and should be viewed only as recommendations. Well, nobody believes that, and nobody acts that way. Everybody out in the field takes these documents as regulation, and that's the way it is. Now, the documents, here's just an example of one, and they're easily obtainable on the web if you want to look at them. What happens is the FDA will put out a draft guidance. It will solicit comments from stakeholders, and then it will finalize the guidance usually in about a year or so, hopefully addressing the comments that have been submitted. So the first of these, which mainly affected manufacturers, but it was the first time that one of these affected labs directly, was on analyte-specific reagents, which came out in 2007. Now, you may recall that an analyte-specific reagent is basically an ingredient of a laboratory-developed test that is sold by a manufacturer to a laboratory. And that ingredient has specificity. So it is an antibody or a nucleic acid sequence or some protein that's binding with a specific target or something of that sort. And what this guidance said to manufacturers was, you have to sell this, if you're going to sell this as an ASR, which means that in most cases you won't have to jump through the FDA regulatory hoops, you have to sell it separately with no other ingredients, and it can't be packaged. It can't be included, no instructions can be included with it, and it can be sold only to laboratories that are certified under CLIA to do high complexity testing. But there was a requirement there for laboratories, which was that if as a laboratory you do a test with an ASR, you have to put this disclaimer on the report. The test was developed and its performance characteristics determined by the lab. It has not been cleared or approved by the FDA. And CAP recommended additional wording that FDA does not require this test to go through pre-market review, et cetera. So that's the first guidance, sort of FDA's first foray into the area of laboratory developed testing. One point that we've seen some confusion with is that this disclaimer is only required for a laboratory developed test that uses an ASR. If the test does not use an ASR, then the FDA required disclaimer is not applicable. The second guidance, which came out in July of 2007, was on in vitro multivariate index assays, IVD-MIAs. And these are assays in which multiple laboratory test results are taken and plugged into an algorithm or an equation which crunches the numbers and comes out with a single index, a single number, which may be a risk factor for development of a disease or recurrence of a carcinoma or development of metastasis or something of that sort. And what FDA pointed out, which is true, is that these algorithms that are used in IVD-MIAs are proprietary, and it's impossible for an outsider to validate them or to look at them and see whether they work or not. So as a result, even if they're developed as a laboratory developed test and used within a single laboratory, FDA said in this draft guidance, you will have to get FDA clearance or approval to do patient testing with IVD-MIAs. Now, it's interesting that there was kind of a split reaction of the test manufacturers to this guidance. Those manufacturers which had their own labs and used laboratory developed testing were adamantly against it. They stated that introducing this kind of regulation would delay the launch of life-saving tests for the early detection of many cancers by at least 5 to 10 years. Might be a little overstatement, and will deter investments in innovative diagnostics. On the other hand, the manufacturers that were more traditional, that sold test kits, that sell test kits to laboratories, felt the opposite. AdvaMed, which is the Manufacturers Trade Association, most of those members believed that supported this guidance and felt that, that IVD-MIA should be regulated like other tests. Now, there are some FDA-approved IVD-MIAs. I thought I would just mention some of those here. One of the most well-known, Oncotype DX, is not FDA-approved. It's still an LDT. Now, interestingly, this guidance was shelved, quote, shelved by the FDA. In other words, they're not going to finalize it. The draft has been withdrawn. But the reason for that is, since FDA decided it's going to regulate all laboratory-developed tests, didn't see any reason just to have a separate guidance on a subset of those tests. So that's why that has gone away. But that's kind of shows you how the FDA is thinking. The other two guidances more directly affect our laboratories. Both of them were put out in last year, in the middle of 2011. The first is on research use only reagents, RUOs, and IUOs, investigative use only reagents. And these reagents, we use them in tests for which there is no alternative, for rare diseases, et cetera. These reagents are exempt from the FDA approval or clearance process. So manufacturers do not have to go through any of that FDA procedure to market these tests. Now, I spoke to a representative of the FDA about this a number of years ago because I was uncomfortable about it. And I said, is FDA going to go after laboratories for using RUOs when you have no reasonable alternative to do a test? And he said, no, that's not the intent. Our intent would be only if there were, say, a high volume test in which there is some alternative and labs are using an RUO because it's cheaper, more convenient, or some other reason. So I was reassured by that. But this guidance seems to show that FDA's thinking has changed, because the guidance states that, and it's directed at manufacturers, but it states that manufacturers should not sell RUOs to laboratories if the manufacturer knows that the laboratory is using that product in clinical diagnosis. And furthermore, the manufacturer should police the laboratory. If the manufacturer learns that a lab to which it sells its RUO product is using it in clinical diagnosis, it should halt such sales. I have to believe that this guidance, at least in part, was some kind of response to manufacturers, pressure from manufacturers, because manufacturers have gotten exercised about the use of RUOs for patient testing. This was just a quote that appeared in a trade journal a couple of years ago from the representative of a manufacturer who stated that RUO companies that are not FDA registered have an open door to have their products used as diagnostics with no accountability at all to anyone. Again, the concept of accreditation, a CLIA, seems to be missing. The RUO-only company has an advantage in that it does not have to spend money or other resources to meet FDA standards. Well, the CAP sent a detailed commentary into the FDA about this, as did other organizations, and pointed out that we have to use RUOs in certain cases where reasonable substitutes are not available. For instance, flow cytometry. A lot of the reagents used in flow are RUOs. Immunohistochemistry, some reagents are RUOs. I believe in HLA testing, a lot of microbiology tests involve RUOs. Diagnosis of rare diseases. And another one, which I admit I wasn't aware of, is that the pregnancy-associated plasma protein A test relies on an RUO, and that for Downs screening, prenatal Downs screening, and there is no alternative to that at this time. So what CAP asked FDA to do is to adopt enforcement discretion towards these uses of RUOs. Whether or not FDA has not responded to us yet about that. Whether or not that's going to happen, we don't know, although in verbal discussions, FDA officials have said they recognize this issue and they understand it, but then I don't understand why this particular guidance was issued. So anyway, we just have to wait and see what they're going to say. The last guidance I want to mention, similar problem, involves in vitro companion diagnostic devices, and this came out also in July of 2011. And what we mean by an in vitro companion diagnostic device is simply a test that determines whether or not a patient is eligible for a specific therapy. So we see a number of those, particularly in cancer these days. HER2 testing for trastuzumab in patients with breast cancer, ALK testing in lung adenocarcinoma, BRAF testing in melanoma, all examples of what FDA would call companion diagnostics. And FDA made the fairly obvious statement that a companion diagnostic device with inadequate performance characteristics could expose a patient to risk, and that therefore all IVD companion diagnostic devices should be approved or cleared by FDA. In other words, no laboratory developed tests allowed. Obviously, this would be a big problem for laboratories, and CAP again submitted comments that kind of like saying, we resent the implication that LDTs have unproven performance characteristics. And once again went through the litany that, we are regulated by CLIA, we're accredited, et cetera, et cetera. And that this would disrupt patient care. In fact, if you look at one example, which is a technicality, but it's one of the problems is, the CAP ASCO guidelines for HER2 testing and breast carcinoma, those guidelines actually require the use of a laboratory-developed test because they've changed the criterion for a positive immunohistochemistry result from what it was in the FDA-approved test, because it was felt that there would be more specific. So actually, you could not follow that guideline based on this FDA guidance. So CAP used some unusually strong language in its comments that this literal adherence to this guidance would subvert a long-standing tradition of advancement in medicine. And again, we have not heard from FDA about this yet, but CAP is maintaining an ongoing dialogue with the agency. Now, so far, actually, FDA has not really done too much in terms of enforcement in the area of laboratory developed testing. What it has done, it has gone after some manufacturers who do direct to consumer testing, some genetics laboratories. It has gone after at least one manufacturer who has been producing an RUO. I don't know the details on that. And it did go after these two laboratories I have up here, these two organizations, because what they did was they said that these tests were laboratory developed tests, but in fact they did not fit the definition because they were being used in more than one laboratory. But I'm not aware that FDA has gone after a hospital or an independent or academic laboratory at this time. Now, how about the future FDA regulations? We don't know the details, but FDA has given us some idea of what's going to happen. FDA is going to put out 3 guidance documents, and they're going to come out sometime in the next year and a half. There'll be one on the overall regulatory framework, one on a registry, which means we'll have to register all our laboratory developed tests with FDA, and one on the description of how the FDA requirements will interact with CLIA. So they'll come out in about a year, and then we'll have probably a year or so to provide comments to FDA with final guidance coming out in 2 to 3 years. And I have to say this slide is actually slightly inaccurate. These are not really new regulations that are coming out. They're these guidances, which are not regulations. And it's easier for FDA to put out the guidances. And in the guidances, if they put out the guidances, they don't have to respond to every comment that we or a stakeholder might make. Whereas if you put out new regulations, the government agency has to respond to every comment. So we kind of think FDA is probably deliberately doing this. Now, of course, we have a number of questions about what's going to happen with these regulations. What's going to happen with the more traditional laboratory developed tests that we talked about earlier? Are they going to be subject to onerous validation requirements? What's going to happen, as we just talked about with the HER2 example, what's going to happen if you modify an FDA-approved test? If you make any modification, if you change the collection device to one that's not listed in the package insert, that becomes a laboratory-developed test. Are we going to have to jump through a bunch of hoops to do that? And so is there going to be grandfathering? Well, FDA has indicated there probably will be grandfathering. It may not be that date in 2003 that CAP had put down. It might be more recent. We don't know. Probably the initial implementation will just focus on high-risk tests. So probably at least initially, this is the idea we're getting, is FDA will not be looking at these more traditional laboratory-developed tests. But we don't know what's going to happen down the line. Now, FDA is looking at having a 3-tiered risk classification. But the problem with this is that it's cramming an awful lot of the tests into the high-risk category. Any test that's a companion diagnostic or involved in cancer diagnosis involving communicable diseases is going to be considered high risk, so that they're going to have more onerous regulations. So that's a concern to us. And I just want to point out very briefly, when we talk about getting FDA approval, it's not a trivial process. That right now, for a company to bring a test to market to sell to laboratories or wants to get FDA approval, it takes a year to a year and a half and costs $5 to $10 million for that company. Now, I don't think it's going to be that onerous for laboratories, but we're probably looking at something significantly greater than what we have to do now. Well, in the interest of time, I'm going to skip over this alternative proposal, which is probably not going to go anywhere politically, and talk about the CAP approach. CAP has been having meetings with FDA since considerably before FDA determined that it was going to regulate laboratory-developed tests. And what we have done is proposed a partnership with FDA, similar to the way we work with CLIA and CMS in terms of accrediting and regulating this type of testing. And the way it would work is that we would use a 3-tiered risk-based system. The laboratory would apply risk, a certain risk level, to a new test, and the accreditor, which might be CAP or it might be some other accreditor, would confirm that risk level. And then the way the laboratory would get the test approved would depend on the risk. So CAP, this is an example, and this is obviously in draft form, of CAP's risk classification, which is a bit different from FDA's. Low-risk tests in this classification would be those which are used with other information. In other words, a test does not stand alone in making a prognostic determination or a therapeutic decision. And for those types of testing, the change, it wouldn't be that much different than it is now. The lab would just have to inform CAP or the accreditor of the risk level, which would be confirmed by the accreditor, and then the lab would go ahead and do its validations and would be inspected at the time of the on-site inspection as usual. The moderately complex tests would be those in which the test often, although not necessarily always, either stands alone as a predictor of prognosis or determines therapy, like a companion diagnostic. For those types of tests, the laboratory would need to submit its validation studies to the accreditor before it could put the test online. The accreditor would approve those studies. The test would presumably go online. The FDA would be informed but would not be directly involved. And the high-risk tests would be those that use a proprietary or non-transparent algorithm, IVD-MIAs, for instance. In those types of situations, the laboratory would need to submit the validation data to FDA, and FDA would have to approve it before the test could go online. Then there would be the usual inspection every 2 years by the accreditor. In terms of the actual validation requirements themselves that CAP is considering, they will be similar to the ones I talked about that we currently have, but there probably will be some more detail. There may be some more specificity in terms of the minimum number of samples. There may be some more information that will be required, but this is just being developed now. It's actually being piloted in a number of laboratories to see how it'll work out. So we want to come up with something which is practical for laboratories. It's not too onerous, and that yet will be acceptable to FDA. So that basically concludes my presentation. What I would ask of you is, if additional FDA guidances come out, the more people they hear from, the more stakeholders, the better. You can see how the manufacturers probably have influenced them over the years by their input. So I would urge all of you, when you hear about such a guidance coming out, and you'll hear about it, sure enough, if appropriate, please send in your comments to FDA, because CAP will do it, but it carries a lot of weight when individual laboratories and practitioners send their comments into FDA. So thank you very much, and I can take questions now, comments.
So Steve, is CAP coordinated with other organizations, and I'm thinking particularly of the Association for Molecular Pathology, in terms of sort of providing input to the FDA, or sort of making sure everyone's on the same page and trying to have some sort of coordinated effort in this regard?
Yes, the question is CAP coordinating with other organizations, particularly the Association of Molecular Pathology? The answer is yes. Actually, CAP is working very closely with AMP. In fact, many of our members sit on both committees on both, so the answer is yes. And if you look at the AMP's comments, which were submitted to FDA, you can kind of tell that the same people were involved with those as in CAP's. So the answer is yes, we are. That's very important. Yes, sir.
You talked a little bit about guidance versus regulation, and that FDA is not really following the rules, if you will, in making guidances. They're pretending that guidances are rules. There was recently the Frank's Lab versus FDA ruling, which basically placed the enforcement of guidance into, I think, a really unknown field. Is the FDA really going to be able to enforce guidances given Frank's Lab ruling?
I'm not specifically familiar with that. Let me back up. The comment was about guidance versus regulations, and is FDA going to be able to continue to use guidances sort of as a regulation? The answer is I don't know. I do know that there was a letter written by a number of congressmen to FDA questioning its approach to laboratory-developed tests. And one of the points that was made was that the FDA was subverting the rulemaking process by going through guidances. And FDA was supposed to respond, and the letter requested a response from FDA by yesterday. And I have not seen that yet. So there is a possibility that So there is a possibility that FDA might be forced to turn to the rulemaking process, but I don't know whether that's going to happen or not at this point. Yes?
Outside of the plan that you mentioned where the FDA would maybe collect a registry of lab-developed tests, do they actually have the resources to do anything more meaningful in terms of inspecting all of us at some point in parallel to a CAP? Do they have any resources to do more than just rely on CAP results or inspections? Because it seems to me like the problem is vast for them to, there's lots and lots of LDTs.
The question is, does FDA have the resources to handle all these LDTs under this new regime? The answer is that they may not, but FDA would not do the inspection. They would not do inspections. They seem to be confident that they can do it. I don't know how. They probably would be relying on CMS and CLIA. Is most likely what they would do. And CLIA in turn would be relying on accreditation. So I think that's probably the most likely scenario. And the other point is that FDA will probably concentrate initially on they will select a few high-risk tests, genetic tests, and go after those and leave the others alone, but they will eventually get to the others.
Following up on that, I've heard it argued before that maybe focusing on the very, very high complexity few genetic tests may be misguided because it's probably more large volume, less complex things that maybe can cause more harm in aggregate if done poorly. Because you may allow, by generating 1 million bad results, you may enter too many people mistakenly into the medical process where they then may get ruled out, but nonetheless they'll have a lot more care. Whereas if you prevent 500 very complex wrong results, that's still unlikely that they're going to happen because people who are doing those extremely complex tests are usually, there are not a lot of big errors done. Can you comment on that?
Yeah. The comment was that there could be a higher risk from a less complex test that's tested on many people as opposed to a more complex test that's just tested on a few. I have not seen anyone address that point. That's a good point, but I have not seen FDA or anyone address that. Yes?
Probably cynical enough to believe that this would never go anywhere, but it seems like a pleasing way to describe the validity of tests is to actually publish the validation. Has anybody floated the idea that validation of laboratory tests should be in the form of a standard method validation format that should be published in, say, a publicly accessible journal, and that it will allow the community to collectively pass judgment on whether the test is valid?
The question was really twofold. Has there been consideration for a standardized way to validate tests and to publish it so that the public can be aware of the laboratory's validation? There has been some discussion of standardizing the validation process as much as possible, and that comes out of this pilot study. I have not heard discussion about publicizing those results yet, although I think that's an idea worth considering, but I have not heard anyone talk about that. Yes, that's right. That's right. Mark.
Do you know what's happening on the international scene? Do other developed countries have similar moves towards greater regulation, and where does the United States fit into the international scene?
The question is, where does the United States fit into the international scene in terms of the amount of regulation? I'm not a real expert in international, but my understanding is that we are far more regulated than most other countries, far more regulated. There's ISO and the CE mark and so forth, but regulation is a far more onerous process in this country than elsewhere. Okay, well, if there are no more questions or comments, thank you all very much.